In this video, we are going to discuss about principles and enzymes involved in biotechnology. There are different enzymes for biotechnology. Suppose the lysing enzymes, the cleaving enzymes, the joining enzymes, the removing enzymes, all are discussed in this video. If after watching the video, if you found that this video is useful for you, then do some needful one for this video. What is the definition of biotechnology? Bio means, you all know, life. Okay, that means we have to use some living organisms, some life forms, specifically the microbes and the biological system, okay, to develop technology and products. Okay, we have to use biological system and the microbes, and they are the living organisms, to develop some technology and products, or we have to modify some products or system for the welfare of human being, is known as biotechnology. And there is another term, genetic engineering. It is the direct manipulation of an organism's gene by using biological system and microbes, known as genetic engineering. And what is the basic principle of biotechnology? The basic principle of biotechnology is to introduce, first to isolate and to introduce the desired gene into the organisms without introducing the undesirable gene. Actually, the basic principle is we have to isolate and introduce the gene of interest, which is in desire, into the organisms, and to prevent the introduction of undesirable genes. This is the basic principle of biotechnology. And the steps involved in this principle are development of recombinant DNA, cloning of desired gene, and then we have to transfer the cloned gene into the suitable organisms. These are the fundamental principles of biotechnology or genetic engineering. This is the pig DNA. We allowed the restriction enzymes. We isolated the desired segment of DNA. We inserted the desired segment of DNA into the plasmid, that is the vector, and in presence of enzyme ligase, there is formation of recombinant DNA plasmid, and the recombinant DNA plasmid is introduced into the host cell. The host is the Escherichia coli or E. coli. Then the host cell is allowed for replication in order to get more copy number. This is the basic principle of biotechnology. For this, we need some tools. Now we will discuss about some enzymatic tools of biotechnology, or the enzymes involved in biotechnology. First of all, the enzyme needed are lysozymes. The first group of enzymes used in genetic engineering or biotechnology are lysing enzymes. Okay, as the name indicates, lysing. Lysis means actually breakdown. The enzymes used to break down the outer boundary of the cell or cell membrane. Okay, now you see here the intact cell. Okay, here it is the intact cell, After allowing the lysing enzymes, the enzyme lysed the cell membrane, and after that the inner content or the intracellular materials released outside, and they are known as lysates. And the intracellular materials, they may be DNA, RNA, protein, or organelles from a cell. This is the main function of lysing enzymes. Okay, when we applied lysing enzymes, then the cell opens up. Actually, the lysing enzymes opens the cell. And some lysing enzymes are lysozymes. They are used for bacterial cell, cellulase. They are used for plant cell, and another groups of lysozymes are there, chitinase. They are used for fungi. Okay, these are the different lysing enzymes to break down the cell membrane. We discussed about lysing enzymes, and the second group of enzymes are known as cleaving enzymes. That means they cleave the genetic material, that is DNA. Okay, cleaving enzymes are of different types: exonuclease, endonuclease, and restriction endonuclease. Okay, this is the DNA template. When we apply exonuclease, actually exo means outside. Here outside means the terminal end, of DNA. The terminal end may be the 5 prime or 3 prime. Okay, this exonuclease acts only on these terminal sites. Now the endonuclease, when we apply or allow endonuclease to the template DNA, it makes a cut in between the middle site. Okay, except the terminal sites, endonuclease acts on DNA. Okay, now actually question is that do they cut hydrogen bonds? No, the enzymes used in cutting process of DNA, they don't cut the hydrogen bond. Okay, what they cleave? Actually, what they cut? They cut the phosphodiester backbone present on DNA. Okay, they don't cut hydrogen bond, they only cut the phosphodiester bond. And third question regarding the cleaving enzymes is, which one cleaves circular DNA? You have read about exonuclease, endonuclease, and restriction endonuclease. Okay, which group of enzymes cleave circular DNA? Okay, your endonuclease group of enzymes, they cleave only circular DNA. Okay, now this is the action of endonuclease, exonuclease, and restriction endonuclease. You see here, endonuclease, it cleave in the middle of the DNA. Okay, here it is the cleaving portion or cut portion. It is taking place in middle of the DNA. Okay, and this endonuclease catalyzes the hydrolysis of phosphodiester backbone. Okay, sugar phosphate backbone is there, and the bond present there is known as phosphodiester bond. Actually, the endonuclease and the exonuclease, they cut the phosphodiester bond of DNA. Okay, now this endonuclease works on circular DNA. And other group is exonuclease. You see here the exonuclease removes the terminal part. Okay, here this is the original DNA strand, and when we applied exonuclease, this exonuclease removes either from 5 prime or 3 prime end. Here this exonuclease removes the nucleotide sequences from the 5 prime end. Okay, this is the removal part, cut part. Okay, now the restriction endonuclease, these are the enzymes, and they also made their cut in the middle of the DNA. Okay, here the cut sites are here. This is all about your exonuclease and endonuclease. Where is the phosphodiester bond? Now you see your DNA is double helical DNA. There is 5 prime and 3 prime end in each strand, and this red portion is known as sugar phosphate backbone. This sugar phosphate backbone contains phosphodiester bond. Actually, the restriction endonuclease, and exonuclease groups of enzymes, they cut this sugar phosphate backbone at this portion, the sugar phosphate backbone. These are the hydrogen bonds. Okay, G pairs with C, A pairs with T. Okay, these are the hydrogen bonds, and they don't actually cut or act on hydrogen bonds. They only cut the phosphodiester bond present in sugar phosphate backbone of DNA. Okay, and the exonuclease, it breaks either in 3 prime end terminal or in 5 prime end terminal. Okay. Another groups are restriction endonuclease, and these group of enzymes were discovered by Hamilton Smith and Daniel Nathans in 1970. You remember this year and name of scientist? And the restriction endonucleases are known as molecular scissors or molecular cutters, as they cut the genetic material. So they are otherwise known as molecular scissors. Actually, the question is, why are they named as restriction enzyme? Because they restrict the digestion of host DNA. Okay, how? By adding methyl group. By adding methyl group, they restrict the digestion of host DNA. So they are named as restriction enzymes. What they cut in your DNA, you all know. I have already said they cut the phosphodiester bond. On which site? This is the important question. On which site do they cut? Actually, they cut in recognition site or in restriction site, and these sites must have some unique characters, and the unique character is the site must be in palindromic sequence. Okay, the site is known as palindromic site or the recognition site on which they will cut. They must have palindromic sequences. Actually, you see here AMADAM. Again, you read in reverse direction AMADAM. When you will found such type of nucleotide sequences in DNA, they are known as palindromic sequences. You see here, actually the palindromic sequences maintain some orientation. If I will read these sequences in 5' to 3' direction, again I have to read in same direction or in same orientation in another strand of DNA. I read in first strand from 5' to 3' direction, and it was GAATTC. Similarly, in another strand, I will also read in 5' to 3' direction, that is GAATTC. These type of sequences are known as palindromic sequences. In that sequence, the restriction endonuclease will cut. How will we give name to restriction enzyme? There is basic principle. Suppose Hind 3 is an example of restriction endonuclease. Okay, H derived from the first letter of genus Haemophilus. Okay, in derived from first two letter of the species. Here Haemophilus influenza is the microbe. H derived from the first letter of name of genus, and in derived from the first two letter of the species, and D is the name of strain, and 3 that is indicated in Roman numeral. It is the order of isolation. Suppose I isolated Hind first, it would be Hind 1. If it is in I isolated second, it is Hind 2. Similarly, Hind 3 means order of isolation. Okay, this is the naming of restriction endonuclease. See here the naming of other restriction enzymes: EcoR1, Hind 3, BAM-H1. You see here genus Escherichia, E derived from Escherichia, CO from species coli, R for name of strain, and I for order of isolation. Okay, and the recognition site for EcoR1 is GAATTC, and it will cut after G. Okay, similarly in Hind 3, H from Haemophilus, IN from influenza, and D for strain, 3 for order of isolation, and the recognition site is AAGCTT, and the cutting portion is the action of Hind 3 is after A. Okay, this is from 5' to 3' orientation. All are in 5' to 3' orientation. Okay, BAM-H1, B from Bacillus, AM for amylo, H is for strain, and I is order of isolation, and the recognition site is GGATGC, and the cutting site is after G. Okay, all are in 5' to 3' direction in DNA strand. There are 3 different types of restriction endonuclease. Okay, basically we are focusing on type 2 restriction endonuclease for genetic engineering. Why we are focusing mainly on type 2? There are some properties of type 1, type 2 and type 3 restriction endonuclease, and the main property is that the type 2 cut both strands at a specific site, usually within the palindromic recognition site, that is 4 to 8 base pair. This is the unique character of type 2 restriction endonuclease. If we consider the recognition site for type 1, actually it cuts but more than 1000 base pair away from the recognition site. If we consider the recognition site for type 3, it cuts the recognition site but 24 to 26 base pair downstream of 3' recognition site. That means type 1 and type 3 they give the recognition or desired gene with some multiple numbers of other bases, but type 2 usually gives the actual gene of interest by cutting in specific sites on the palindromic recognition sequence. Okay, so type 2 is essential for genetic engineering, and other properties are also here. All require magnesium ion. You see here, all require magnesium ion, and the nucleus subunit structures are there. Type 2 is a homodimer. You have to remember that type 2 is a homodimer, and type 2 has a single recognition site. This is the most important one. Single recognition site is for type 2, and the site of methylation actually takes place at the recognition site. Okay, everywhere the methylation is taking place at recognition site, but in type 2 it is also operating in recognition site. Okay, if we consider some restriction and modification property, they have separate nuclease and methylase property. It is for type 2, and other properties are there. All properties are different from others. Now the types of cut produced by restriction endonuclease. Okay, actually two types of cuts are there. One is sticky end cut. You see the cut portion is asymmetric one, and second one is blunt cut. That is the cut takes place at a symmetry. Here the symmetric cut, here the cut portion is asymmetric one. The asymmetric cut is known as sticky end cut, and the symmetric cut is known as blunt end cut. These are the two types of cut produced by restriction endonucleases. Endonucleases which we are using in genetic engineering. The sticky end cut. Okay, actually they get their name because they have overlaps. You see here they have overlaps. You have seen in previous figure the overlapping portions are there. Okay, and the restriction enzyme cleaves it asymmetrically. Okay, asymmetric cut is there. You see here, this is the normal DNA. When you allow echo R1, it digests the DNA and produces sticky end cut. There is overhang. Okay, there is overhang is here, and this is known as two types of overhangs are there. One is 5' overhang and another one is 3' overhang. Okay, in 5' overhang there is overhang of 3' hydroxyl group, and in 5' overhang there is overhang of phosphate group. Okay, and the second cut is blunt end cut. Echo R5, when you allow this enzyme to digest the DNA, it produces no overlap cut. It produces a symmetric cut. This is known as blunt end cut. And these are the two types of cut produced by restriction endonuclease. This is the intact DNA and remarked by the cutting site, cut site. Here in figure 1 and 2, you see here the cut site is in asymmetric one, the asymmetric cut site, not in symmetry. In figure 3, the cut site is in symmetry. Okay, when we allow hind 3 to digest this DNA, it will produce 5 prime protruding end. That means 5 prime overhang. 5 prime overhang means the overhang is marked by phosphate group. When we allow PST1 to digest the intact DNA, it also produces 3 prime overhangs. Okay, 3 prime overhangs means 3 prime end containing hydroxyl group, the OH group. Okay, and eco R5, when it digests the DNA, it produces blunt end. There is no overhang, produces blunt end. These are the different cut portions of the DNA by allowing different enzymes. Here 5 prime overhang end, 3 prime overhang end. These are the asymmetric cut, the sticky cut, and this is the blunt end cut. Okay. Some enzymes and the source where we obtain them, and the recognition site and the nature of cut produced by the enzymes. Actually, the enzymes EcoR1, Hind3, BAM H1 and SAL1, they produce sticky cut. You all must have to remember the recognition site and cleavage site of these enzymes, because these enzymes produces sticky end cut. Okay, and other enzymes produce blunt end cut. Okay, if you consider EcoR1, E for Escherichia, C for coli, R for RY strain 13. Okay, Hind 3, here you all have studied about the naming few minutes back. These are the enzymes, the sources, the recognition and the cleavage site, and you have to remember the after base pair cutting site or cleavage site in which direction. Here I am telling GAATTC. It is the recognition site of EcoR1. Okay, and it is in 5 prime to 3 prime orientation, and the cut site is after G in 5 prime to 3 prime orientation in both strands. Okay, you see here after G in 5 prime to 3 prime direction. Similarly, for Hind 3, the recognition site is AAGCTT in 5 prime to 3 prime direction, and the cut site is after A in 5 prime to 3 prime orientation. Okay, you see here SMA1, the recognition site is CCCGGG. It is in 5 prime to 3 prime direction, but the cut site is after C in 5 prime to 3 prime direction. You all have to remember this one. Okay, this table is the most important and informative table regarding the enzymes, their sources, their recognition and cleavage site, and nature of cut ends. Now, the action of restriction endonuclease. The action of restriction endonuclease, you see here, the enzyme cuts both DNA strands at the same site. Okay, the recognition sequence you see here and the site you see here, and this is the vector DNA and this is the foreign DNA. You allowed your EcoR1 to cut the DNA, and this after allowing EcoR1 you found a sticky end cut. Okay, and the sticky end DNA fragments join to form a recombinant DNA. This is the action of restriction endonuclease. Another group of enzyme is DNA ligase. It is known as gap filling enzyme or joining enzyme. So it is otherwise called as molecular glue or molecular glove or genetic gum, as it joins the fragmented portion, so it is known as joining enzyme. It fills the gap between two DNA fragments on the phosphodiester bond. Okay, you saw the action of restriction endonuclease to form a recombinant DNA. This joining was carried out by DNA ligase. You see here, this is the cutting portion, this is the sticky end cut, and you have allowed ligase, and ligase seals the gap. There is no gap. So DNA ligase is known as joining enzyme or gap filling enzyme. Other group of enzymes are alkaline phosphatases. Phosphatases are the enzymes. They remove phosphate group from the 5' end of vector DNA, but not from the desired segment of gene of interest. They only remove phosphate group from the vector DNA at 5' end. Actually, why the treatment of alkaline phosphatase is necessary? Because to prevent self-ligation. To prevent self-ligation, we will allow alkaline phosphatase. What is self-ligation? Okay, here you see in the figure, in point 1, the double stranded DNA, circular DNA is here. It is the plasmid DNA. Okay, double stranded circular. After cutting the double stranded circular DNA, there is the 5' phosphate group and 3' hydroxyl group. If we will allow this cutting as such, there is possibility or they will ligate itself and form a complete double stranded circular DNA. This is known as self-ligation process, and this process operates in presence of enzyme ligase. In order to prevent this step, we have to allow phosphatase. Okay, now this phosphatase will remove the phosphate group from the 5' end. Here there is removal of phosphate group from the 5' end, and there is presence of hydroxyl group at both 5' and 3' end of the coated plasmid or coated vector DNA. And by allowing ligase enzyme, the self-ligation is prevented because there is no phosphate group. There is no phosphate group at 5' end. To this vector DNA, we will allow our gene of interest with enzyme ligase, and this will form a recombinant DNA. Okay, now we treated the alkaline phosphatase to this system in order to prevent self-ligation. Here, after treatment of phosphatase, self-ligation process is prevented. Other group of enzymes are reverse transcriptase. As the name indicates, they form the DNA copy using RNA as template. Okay, and they are found in retroviruses. Actually, they use RNA to form DNA. From RNA, there is formation of cDNA and then double stranded DNA. Okay, and this enzyme is otherwise called as RNA dependent DNA polymerase. DNA polymerase enzyme that transcribed single stranded RNA into double stranded DNA. They are known as reverse transcriptase.